Okay, gang, here's your video on section 2 of chapter 11. This is specifically about the reception of a signal, and the first picture I have up here is the picture that I ended the last video with. It just talks about when we're communicating, two cells communicating, typically on the receiving the signal cell, there's three steps that are involved in a cell receiving a signal and then actually doing something with the signal that they have received. So there's the reception. First step is the reception of the signal, or receiving the signal. The second one is transduction, and then the third step is activation. So this entire video here is focused on the receiving of the signal, or the reception stage here. So first of all, we can take that and fit it in the whole receiving or reception, have that fit into two different classes. The first would be types that are signals that are received inside the cell, or intracellular receptions, and then the second type are ones that are on the surface of the cell or in the membranes of the cell, which we'll get to in the next slide here. But intracellular reception, the example they use here is testosterone. And first of all, the definition of intracellular reception, all you need to know there is that the signal is received by a receptor protein inside of the cell. Okay. Now, specifically the example that they're giving, if you remember, I had said that all these types of communication are going to be different with every single communication signal that is sent from one cell to another. It's always going to be responded to differently for every different scenario. So this happens to be the one that they're choosing to focus in on for the intracellular reception, and it is dealing with testosterone, which I talked about in the last video. But testosterone is a very hydrophobic molecule, and so it actually passes through cell membranes really easily, if you remember those characteristics of cell membranes. But so small particles can do this as well, but testosterone and a lot of other hormones actually are able to just pass directly through cell membranes, and in doing so they can enter any cell in the body. When they get in there, if it's a cell that happens to have a very specific receptor protein that recognizes testosterone, it will link to that, and in doing so it just activated that receptor protein, and then action can happen. In the case of testosterone, what it does, which is a really unique thing, but when the receptor protein and the testosterone join, they actually go into the nucleus of the cell, and they start to act as something called a transcription factor. Anyway, a transcription factor basically turns on a section of DNA and says, hey, use me to do something, okay, to make a protein, which is what DNA does. And so this will actually go and link up to a section of DNA and basically turn on a gene. And turning on that gene, the whole process of making a protein happens. And so in the case of testosterone, these genes that they turn on are the genes that lead to male characteristics, male sex characteristics. Anyway, and that's through that system. So that's intracellular, simply because this molecule actually attaches to a receptor protein that's already inside of a cell. And this one ends up being a very simple process. Testosterone and how it works is a very simple process. Wanted to introduce a word that the book, as I was looking through here, the book says that it doesn't really define well, and I don't know that it's shown up at all this year. There's a lot of words that haven't shown up at all this year, but anyway, it's ligand, L-I-G-A-N-D, ligand, and basically, I don't know why they decided to throw a different term in here, but a ligand is basically a substrate, okay, on an enzyme. A similar idea. A ligand is something that very specifically is recognized by a protein and fits into basically the active site of a protein. The reason it's not, I do know the reason they don't call it a substrate, is in this case it's not actually attaching to an enzyme, it's attaching to a protein, and so they don't call it a substrate unless it's actually an enzyme that it's attaching to. But it's the same concept. We would call it a substrate, or a ligand is what you're going to see several times in the book. So, alright, we'll get on to the receptors that are stuck on the surface of a cell next. Okay, so we're going to look at the receiving systems that are stuck on the plasma membrane, and these would be for molecules that are either too large or are hydrophilic and are not able to go through a cell membrane to get the signal to the inside of the cell. So basically the goal with these membrane located signal receivers is that they're trying to get the signal from the outside of the membrane, get it to the inside of the membrane where it can actually do something. And so this is where the chapter, I think, starts to get, I don't want to say confusing. It's just, you could easily read an entire section, and when you're done, just be like, what in the world did I just read? Anyway, and it's because it gets pretty heavy on terminology at this point, so hopefully I can simplify it a little bit for you. I feel like I'm just saying what's in the book anyway, but hopefully I can simplify it a little bit. But anyway, the first type here is called G-protein linked membrane receptors, and basically they are integral proteins that are on the membrane of the cell, and integral means that they go through the entire membrane. And there's a picture up here. It shows there's some alpha helices in there, which is good review from protein structure. But anyway, there's two locations on this that are of interest. There's one on the outside, which is where the signal, whatever signal is being received, where that signal is indeed received. And then there's a location on the inside that's going to interact with something called a G protein. And so we have this receptor here, and then our G protein. The G protein is a peripheral protein, which just means it stays on the outside edge of the plasma membrane, and it can move around there. So it can actually move around on the inside edge of the plasma membrane. And this G protein can either be active or inactive. The difference between the two is depending on which molecule it has on it. There's two options. It can have GDP or GTP. These two molecules are very similar to ADP and ATP. It's guanine diphosphate and guanine triphosphate. Very similar in structure. Anyway, if GDP, if that form of it happens to be on the G protein, it is not active. And if GTP is on there, then it is active. Those are two options. So once our G protein receptor, once it receives its signal, whatever that happens to be, then that activates the inside of the protein, that site on the inside, and makes it an active enzyme, and the reaction that it is going to facilitate is going to be the removal of the GDP, something we don't want, and we're going to place GTP on this G protein, and that activates it. So once that has happened, once that reaction has happened, then the activated G protein is just going to be released from the receptor protein, and remember, it can move around in the plasma membrane. Actually, all these proteins can move around, but anyway, it'll move around until it reaches some form of an enzyme. And again, this is totally dependent on what reaction is needed and what response system we're set up on. But anyway, this G protein will find the particular enzyme that it can match up with, and now that it's activated, it will be recognized in the active site of the enzyme, and it will activate that enzyme. So once the GTP, or the active G protein, goes into the active site, then that enzyme can do whatever it's supposed to do, which is pretty broad statement there. But whatever it's supposed to do, we just activated it, and it can cause that to happen. Now, the G protein itself, once it has activated an enzyme, it will, most of the time, it'll go through the hydrolysis of GTP, where it'll knock off that phosphate that makes it TP, triphosphate at the end, and it will make it a GDP molecule, and so it will just have inactivated itself or deactivated itself, and so we're back to where we started. At this point, that G protein could be used once again to receive a signal from another activated receptor protein. So that's the G protein linked membrane receptors. That's how they work. Several steps there, but it's pretty simple. One molecule gets turned on, gives the signal to another molecule, which is then turned on, and then that goes and gives the signal to an enzyme, which is then turned on. Okay, so it's just turn on or activate a molecule, pass the activation onto another molecule, pass that activation onto another molecule is what we're seeing on this one right now. So that's G protein linked membrane receptors. The next type of membrane-bound receptor system is called receptor tyrosine kinases. First of all, in the name there, tyrosine is an amino acid. It's one of a specific amino acid. And then a kinase is anything that adds phosphates onto something else. It's a kinase. If you remember, phosphofructokinase was one of the that was one of the enzymes, an important enzyme in glycolysis. But anyway, with this, the diagram they're showing here, it's another integral protein, goes through the entire membrane. It has a receptor on the outside, and then it basically has an enzyme on the inside, or an active enzyme site on the inside of the membrane. And these work, two of these together, and when they both get activated, or when they both receive a signal molecule, then they will end up bonding together. They call it dimerization. A dimer is typically when you have two of the same molecule that are being bound together. We'll hear that again later on with some genetic mutations that happen because of dimerization. But anyway, we get a dimer, and then that's the activated form of these tyrosine kinases. So we've activated these enzymes, and the thing that they're going to put phosphates on is each other, actually. So we've got these two molecules together. The one protein is going to phosphorylate the other one, and the other one's going to phosphorylate the first one. So we get to the end here where there's phosphates on all these different tyrosines. There happens to be 6 in this diagram, but all these different tyrosines on each of the proteins. So we've activated this whole system, we've got it phosphorylated, and we're going to get something called relay proteins, and a term that I have not really used yet, something called a transduction pathway. But basically, what we've done is we've gotten the signal from the outside of the cell to the inside of the cell, but it still has to be carried to wherever activity is going to happen. Typically, it's a series of things that happen that get the signal from the membrane here all the way into wherever the activity has to happen. That series of actions or series of steps that gets it there, that's called a transduction pathway. In this case, what we've done is we've got the signal to the inside of the cell, but now we have to launch this transduction pathway. And so we have these relay proteins that they've marked here. And essentially these relay proteins, they come along attached to the phosphorylated tyrosines, and that activates them so that they can pass the signal onto the transduction pathway. Okay, and so again, we've accomplished our main goal. We've got the signal from the outside of the membrane to the inside of the membrane, so that a transduction pathway can come and take the signal and continue to pass it down the line, which we'll learn about in the next video. So one last type of membrane-bound receptor system here. And this last one is called an ion channel receptors. This is the simplest one, I think. But basically, a signal is received by a gate protein that's on the membrane of the cell, and the signal basically just opens up the gate protein, opening up a pore so that some other molecule can fit through. The signal molecule is not opening this up so that it itself can get through. It's a different molecule here. But often what will happen, oftentimes it's an ion here, that's why it's called an ion channel receptor, but calcium or sodium are examples of that. And what they'll do is they will enter the cell, increase the concentration of themselves on the inside of the cell. A lot of times that'll trigger all kinds of different changes of proteins inside the cell. So if all of a sudden we have a whole lot more calciums inside of a cell than we normally would, then there will be proteins that will actually be activated or deactivated by that. So this signal, once received on the outside, we open this pore and it's going to change the environment on the inside, and that way our signal has actually entered the inside of the cell, because it's going to trigger some proteins to work, and some other proteins not to work. So those are all the different types of receptors that the book goes through. And again, and I'm going to say this a bunch of times, but again, these are just examples. These are some systems, there's other ones, and every single time there's a signal from one cell to another, every different case is going to be just at a different case. They're going to work differently. So anyway, the next video that we're going to get to, now that we've received these signals and somehow gotten them to the inside of the cell, the next step is getting them from they're inside the cell to let's get them to the right place in the cell so that things can happen. In some case, we have to take these signals and amplify them, they call it, or make them a larger amount of signal. Anyway, we'll get into that in the next video. Yeah, we'll see you then.